Yeah, so I already got a question saying that, so in my opinion, do I think the final is harder than the midterms? So in terms of the uh, the finals, it will be definitely harder. And uh, in terms of that it's cumulative and it includes material from the first section as well. And more so, the real killer is that during the final exam period, you're going to have so many different exams to take care of. And so the actual time you can allocate to study for 265, it's slim. So for that very reason, you should assume the final is harder. And also the materials for the second half is also very application based as well, but it also rely on a lot of memorizations as well. So insofar as difficulties is concerned, I would say yes, it's a little bit harder. If, however, yeah, I told my group as well, like, so if you study very hard, okay, so, and I shouldn't say very hard, you study as hard as your midterms, and you should realistically expect anywhere between 80 to 85 for your final exam. Yeah, and so one other things I probably want to point out is that so for final exam, unless Dr. Chang actually posts some old exam questions, there is no officially publicly available old exam questions. So if people, however, through connections, they can get some old exam questions and more powers them. But the officially there is no exam being posted unless Dr. Chang herself posts questions. Yeah. Okay, sounds good. So we're now 315. Let's get started. Okay, so as you know, then we are going to have a study break coming. So wish everyone a happy study break in advance. And second, so as you know, there is actually a written assignment due very soon after the study break. So make sure, please read the instruction carefully and in terms of what kind of a paper you can choose and especially what kind of topics you should write about. Okay, and please get that started as early as possible. And this is an easy 20% or 25% that you can grab and don't let it go. And if you do well on that, this is one item that if you spend more time on it, you will do better. And so time is not an issue. So just spend more time on it and make sure you get good grades on it. So this will elevate your pressure out of the finals a lot. And lastly, of course, sleep and relax and read. So for this week. So again, so I kind of showed my group this philosophy on the left. This is when Mr. Bean actually, after learning, oh, this exam is not really about what I studied. So let's just make sure in terms of finals, make sure this does not happen to you. So if midterms already, this is the case for you. So please make sure you do a thorough midterm reflection. Doesn't matter you did well or not well. Just think about it. Though, did I actually study everything that is going to be tested? And or I studied a bunch of complements to what the exam is testing. So, what strategy I studied works and what didn't work. So this will be some of the things you need to reflect and apply. Cool. So now let's move on. Slides a little bit frozen. It's moving. So yeah, so once again, that's Alex and me, and I guess Alex is the wild type and I'm the mutant, or you can call a different allele here. So we'll see, or because I possess a SNP here, not just a SNP, I have multiple mutations compared to Alex. So we'll really look at the differences between SNP, allele, mutations, and all this stuff, and let's settle those once for all. So once again, so the plan for today is assignment and lectures. So assignment, we already talked a little bit before the slides, but just all in all, I just want to say that so a real well-constructed essays should be really logically connected. So when the person actually reading your essay, read it, and he or she will have a good idea like where this essay will flow to, and as such, it will be a great idea if in the introduction, if it will be a great idea if you actually pick your paper first and then write your introductions and build the introductions as a kind of a suspense as to what is going to happen. And so obviously you're going to introduce this disease, which should have been a monogenic disease. And then you tell people like the genetic cause has been identified. However, there's still so much unknown to it. And then you write specifically about a 1 or 2 area that is unknown. And this will be the area, this will be the gap that's going to be filled by the 2 paper you are about to present. And so where should I get those information about which gap should I fill? Usually the paper that you chose in their introduction, there should be something about it in their introduction. Basically, they dig a hole first and then say, oh, you know what, this hole needs to be filled. And there you go, that's us. We're going to fill this hole. And in the conclusion paragraph, make sure you, again, emphasize this 2 papers' contribution to filling this holes, and why is important this hole being filled into the grand scheme of understanding this disease. So this will be the essay. And I think it should be clear enough according to the introductions of the assignments. So we still actually got a bunch of emails regarding is my paper okay and not okay, and is this a review or a primary articles. And again, I just recommend that we go back to the previous tutorial, previous review sessions, either by Alex or by me, and just see what are the characteristics of a review article and also a primary research article. Perfect. So my very last point I want to make regarding the paper choice is that typically a journal has its kind of a style and also its preference for the content of the paper. So for example, Nature, so without saying, for the biomedical field, the most prestigious papers are basic research papers are Nature, Cell, and Science, those 3. And they are obviously like the big 3s. If you are able to get into those 3 papers, journals, then you're the best. And unfortunately, that's just how it is. And so the question is that those 3 journals have very, very different emphasis. So for example, the Nature and Science, they emphasize more. I'm a little bit being kind of a generalizing and also being against this kind of a top view, but Nature and Science usually they emphasize novelty. You have to be the first and right, and they care, however, less about the mechanism. And Cell Press papers, though, and they care more about the story, and their stories are usually comprehensive. And you read their essays, you read their papers, it's super long, but their stories are really well laid out, and you see a good logic flow throughout. So as such, for your purposes, so I would say a Cell Press journal will be more suitable because they actually tell you a story. And make sure you avoid those journals, those papers that are appearing in the early 2000s. That's when the sequencing era just started boom. And so you definitely don't want papers just are doing only sequencing, and I sequenced 1,000 patients and I got a mutation. And this mutation is associated with a particular disease. And boom, I identified a potential associated gene to do that disease. So you already knew from Belinda's section that association doesn't equal to a causation. And this essay is exactly not about association. They need causations, a type of study. So just keep that in mind. Okay, so I got one question, say, how detailed should our description of the result in the paper be? So that's a great question. So how do we actually, how detailed should we go to? Especially if you look at papers after 2015 and later, those papers are super long, and it's impossible that you can condense everything into one sentence, in one paragraph or so. So there are a few things I can actually recommend you to do, and unofficially, by the way, and is that if you publish a really good paper, if this paper is really worthy of kind of a focusing, and those guys are usually the universities or their whatever institute will try to brag about it. Oh, you know what, Dr. A and B like managed to discover this. So they will have something called a press release. So if you read the press release, they're really written in a layman language, but yet with kind of a nerdiness of a scientist. So your level of writing, I would say, if you kind of align yourself with the level of those press release, you did a great job, because that is easily introducing the concepts, but also to the level of difficulties that the public and also the general reader without a previous background knowledge of the disease would be able to understand that. So in terms of like, what should I focus and what level of details? So you definitely don't need to really go into the exact details of what, so they use the Western blot and show that there was a 10 kilodalton size shift. Those kind of details you don't have, and they use this control and all that, that you don't really need to go that details. But if you read their subheadings of each section, okay, say P53, like MDM2 regulates P53. So they will have a bold font subsection headings. So this will be, if you actually gather all these subheadings together, this will be at the right level of details you should probably get into. So again, those are headings are already very summarizing already, and you should really fill in further with one or two details. Let's say that they use a Western blot to show this. They use a mouse model to conclude this and that. So I hope that makes sense. Perfect. So one question, private question to me says, is it okay if our primary research article is old? I found the original research paper that discovered the gene, but it is from 1995. So yes, so for my section, for my tutorial, again, this is really like unofficial. It's just person to person. So for my sections, I this is my opinion. So if you find old papers, so in terms of like, for you as a writer, it's easy because they're short. So modern days, like easily, old days, I don't know, like 10 papers is, sorry, like 2 or 3 papers together is not even the experimental amount for nowadays 1 paper. So a figure nowadays, it's probably like a few papers in the past. So definitely, if you have a journal articles from modern days, it's more difficult to summarize. And so that then why should we not use old articles then? And or something against using it is because that old technologies are probably constantly evolving, and their conclusions are probably being updated as well. But in terms of in terms of the discovery per se, like there's nothing wrong with using a paper from 1995. Just make sure the paper you chose not only sequenced or in 1995, they're not able to actually sequence them. They're probably going using some other mapping and association method. So just make sure, in addition to that, they actually did some molecular follow-up to that. Then you're good to go. Does that answer the question? Does that answer the question? Yeah, perfect. Thank you. Okay, so now let's look at the very first concept of this week, which is the biosynthetic pathways. Okay, so I just want to kind of quickly gloss through the third slide. So looking at those minus sign and plus signs. So again, so I told my sections on the exam, the final exam, because it's a cumulative exam. So it's very important that like you knew that you have to know both sections material, Naomi and Belinda, together in order to do well in this exam. Knowing that, though, the exam is actually made separately by both of them. So sorry, they do not, I mean, they probably will talk to each other, but when they make the questions, they likely not. So they're pretty independent in making the questions. So yes, there will be integrations of the materials, but likely unintentionally it got integrated. So it's important that you actually knew who this question is actually from. This will make your life so much easier. And one example is that, is it due to additive effect, or is it actually due to dominant, co-dominant, this kind of thing? So just kind of an advance, a warning in advance. So one thing I want to actually also point out is that if you look at the this kind of a minus plus kind of a sign, so just make sure that this is about a biosynthetic pathway. It's not really about Naomi's complementation task groups. Okay, so just kind of a heads up. So let's go back to the original material. So this is a classical biosynthetic pathway. So whereas you started with the initial compound, okay, so I have, I don't know, like Burger King, I have burgers and bread puddings, and also I have those meat pad, and eventually I have number 5. So this is when everything got assembled, and when everything got assembled, you start to actually get the final product. And what drives all this entire step is that at every single step, you have enzymes that actually catalyze this reaction. So every step, you're making the Burger King pad like bigger and fatter and more tastier, and to the eventual effect of number 5, which is your final product. So the question is, though, that at every step, you rely on the enzyme to be fully functional. But if the enzyme is, so the enzyme function is affected by, is controlled by the gene, it's coded by the gene. So corresponding to this ABCD here. So if any step, okay, and you just need one, any step that does not stop to actually work, then you mess up the entire pathway. So basically, College Street, I can just dig a hole in front of St. George, then the entire College Street is doomed. You can't really use the College Street anymore. The only solution to that is that if you were, let's say, in this chart again, so let's say enzyme B, now it's messed up, okay? And so the only product that could have been made is the product that is immediately just before that. So you can make stuff up until number 2 before B actually comes in, okay? So the production of B is still fine, but anything after that is screwed up. You can't really do it anymore, because B is blocking it. You like B that is actually trying, that can function, be functional for it. Then the question is that what is the function of B then in this case? So the B's function is to actually convert product number 2 to number 3, okay? So basically just add onions to the burger. So the question is that if instead of actually relying on enzyme B to add that onion to the burger, I actually just add the onion myself. Okay, so you provide the entire pathway with product number 3 directly. Okay, so this will effectively actually rescue this pathway. In so doing, the pathway can still actually go on, where you can actually continue with enzyme C, which uses product 3 to make product number 4. So this is the basic idea of rescuing a pathway. And as such, the step that can actually rescue a pathway must rescue after the fact where the blockage is. Okay, so your rescue, the product 3 must be, if it were able to rescue the pathway, then it should come after where the blockade is. So suppose I actually add product number 1 to the entire pathway, knowing blockade is actually at enzyme B. It will not work. It will not work because it still got blockade at enzyme number B. Okay, so if I were able to actually add a particular product, let's say number 5 directly, that's not fair. I'm just adding, yes, let's do that. Let's add product number 5 to this entire pathway. Then you will see, I don't care, A, B, C, D enzymes, they can all be messed up because I'm adding the final product, okay? And obviously I'm gonna rescue this pathway, right? And if on the other hand, I just add product number 1, okay? And doesn't matter A, B, C, or D enzyme is non-functional. I can rescue none of them because this is the very first one. So this kind of gives you an idea of like how to study this kind of material. So you always use extreme examples. Okay, so the extreme far less and extreme far more. And so adding 5, the final product, the product that actually can rescue most of the pathways or the mutants. Okay, this will definitely be the latest of the entire tested compound. If a compound is the most useless one because it cannot rescue any of those mutants, or actually the least amount of mutant, then you knew that product must be involved in this pathway in a very early on stage. Okay, so I hope that kind of clarified the question a little bit. And then if I actually interpret this into a real experimental settings, then what's that going to be? So here is an interesting thing. Okay, so right now I have the compound being added, and also I have different mutants. So what does a mutant mean? So we later on we will study what mutation is. So in terms of a mutant is concerned, so we have strains, usually yeast or bacteria or those things that are easily manipulatable, and genetically manipulating them. So we create a mutation on a particular gene, usually a loss of function mutations in those genes. So basically that gene A is no longer functional. Okay, so what's going to happen is that this gene A is no longer backtrack. Gene B is here, so gene B is no longer functional. Then this a mutant strain, this like all the offspring of this mutation of this yeast strain that does not carry this a functional B mutations, they won't be able to actually have a functional enzyme B made. Okay, and the question is that which compound can actually rescue it. So again, this is the example we went through in class, so I'm not going to go over them again, but just to kind of repeat what I just said. So the ones that the compound that has the least, that has the most effects, that can rescue most of the mutant, they must definitely come from the latest. Okay, just my example of adding the final burger to the product line. It's definitely going to rescue everybody. Cool. So without further ado, let's actually look at a question. Okay. Oh, yes. For that, this is the essay. Sorry, the slides examples that you have gone through in class. So make sure you kind of go over them. But let's look at a question here. So I'll give you 2 minutes. I'm just going to shut up and let you to work on it. It's 2 minutes, so time flies, just like the midterm. So let me ask you the first question, which is the first step you should do as well. So can anyone tell me, is the compound ABCD or 1, 2, 3, 4? So which one is the mutant? Which one is the compound? So let's say ABCD. Is the ABCD the compound or the mutant or the genes? Awesome, awesome. Thank you very much. So the ABCDs are the compound. Okay, and obviously this question is very easy, like because it says the mutant and compound numbers are different. Okay, and so that just makes the whole thing way easier. But if I'm just gonna go back again. Whoops. Here. Close. Yes. So yes. So there we go. So the compound, okay, compound is often like here more than the gene itself. So you have N+1, you have gene +1 compounds usually involved in this entire pathway. So the compound is usually more. Cool. So question number 2. Okay, so now we are clear, which is compound. Then tell me which one is the most powerful compound. A. Okay, so what does it tell you that? If it's the most powerful compound, then this dude must be the very last one. Perfect, perfect. Thank you so much. So we're doing very well so far. And who is the most useless one? C. Awesome. So now we get our terminal choices already made. Okay, so CA. Had this been a multiple choice, you probably already get rid of like 1 or 2 of the junk choice already. Okay, so again, this is what happens when had this been a multiple choice exam. Obviously, this is a short answer question, but had this been a multiple choice question and you're short on time, okay, you already kicked out. Like you kind of boost your confidence already by 30% already, 30 to 40% already. Okay, so now without really like kind of think about this or write about it, okay, you can just use this very rule of who has the most, basically based on the powerfulness of the compound, you can kind of already start ranking them already. Okay, so C is the least useful one, then B is only second to that, so B will be your next. Okay, and so F has 2 crosses, F will be the next. Okay, and yada, 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 the same thing. So C, B, F, G, E, D, A. So that indeed is your final answer. C, B, F, D, E, and G, A. So this will be the final answer. Cool. So I hope everyone get this question right. Perfect. So show me, if there's any questions, just feel free to pause me. We only have a few people here, so it's very easy to just turn on your mic. C, B, F, G, E, D, A. Yes, yeah, you're right, Jeff. Yeah, G, E, D, A. Perfect. Okay, so let's move on to the next question. Well, actually, that's all the question, a synthetic compound I have already. Oh, can I explain how I get to the middle ones again? Okay, so no problem. So again, so I just use the trick of like using the powerfulness of ranking. So who is the most powerful ones? Then they are the later. And the less powerful you are, the earlier you are. Okay, and by this very order, you already knew what are the orders. So the middle ones are just the same idea. If you are kind of a less powerful than the other ones, let's say just D and E. Okay, so between E and D, who comes first? So clearly E comes first because E rescues 1 less mutant compared to D. So D should come after E. Does it make sense, Mohammed? Yeah, perfect, perfect. Cool. Okay, so now we have to really now assign, actually let me just type the answers here for the folks that are watching later. So the rest of the crew, we have to start assigning where the mutant actually fit into this. I'm shutting up again, and so I'll give you 1 minute. So please fill the mutant into the charts. Which gene is being so the mutant will carry, sorry, I keep on talking. The mutant is carrying the gene, okay, that is, I'm just gonna group. Okay, so mutant carries the enzyme that will catalyze the conversion between C and B, okay, and the mutant carries mutation in that very gene that probably will fail to do this conversion. So can you assign where the mutant is? Cool. All right. So this is the logic that we actually use exactly the opposite as just now. Okay. Instead of actually looking at the compound where who is the most powerful compound, we look at who is the most stubborn mutant that nobody can rescue you. So what does that tell about the property of this mutant? Should you code for an enzyme that like functions at very early stage or at very late stage? If you are super stubborn, nobody can rescue you. No compound can rescue you. We already got two answers, early and late. Think about it. We're waiting for the third person to vote. What was the question? So the question is that I am a mutant. Okay, so as such, I carry a mutation in a gene that catalyze the conversion of compound. Okay, so if I am a super stubborn mutant, so on the same role, okay, no compound can ever rescue me. So should I be a mutation that is encoding for a gene, okay, in the very early stage or at a very late stage? Aha, so the answer is late. Perfect. Okay, so as such, compared to the stubbornness of this, so here again, so plus means that you are being rescued, okay, and minus just means that you can't be rescued. So as such, number 3, okay, being the most stubborn of the entire panel. I mean, the stubborn, I just made up this term, but like, you knew that kind of the most hopeless ones. And so number 3 should be the latest ones. Okay, so they should code for an enzyme here. And who is the most powerful, who is the most easygoing ones? Number 1, okay. So I'm not going to fill in the rest of the mutations, so you feel free to do that yourself. But the question is that so does it actually make sense here? Let's look at the chart now. Okay, so we look horizontally. So if 1 fits in here, okay, and C, adding compound C, does it actually rescue it? So we know it doesn't. But in reality, does it really make biological sense here? That's indeed, because we add C, which happens before the number 1 comes in, okay? So obviously you can't rescue it. So that's basically the logic of this kind of a question. Okay, show up of hands if you're clear about this concept, and then we can move on. Perfect. Feel free to promptly announce to the public that you're good with the concept. Cool. Okay, so move on with the next question, the next concept, which is mutations. So I hope you guys are okay with this question. So even though it's not part of the questions, and I often like to add a little bit kind of to it. Oh, Jeff says, wait. Oh, you can just turn on the mic for small class right now.
Okay, yeah, so the question doesn't ask about the mutants, right? So we only needed to answer for the compounds.
For question like the ones right now shown here, yes.
Yeah,
the
fast,
B is more, yeah, part B is more
okay.
like impromptu. I just randomly pop it up. Yeah, cool. But are you able to actually solve this based on our logic?
Yeah.
Awesome. So you're all good. Yeah. Cool. Next question. Next concepts. Mutations. Okay. So just for the pleasure, I'm gonna read through the slides a little bit. So mutations occur at all time. Okay. And the question is that a mutation rates, even though it occurs at like so frequently, everywhere, the rate of mutation is actually very, very, very low. Okay, and I can say the mutation is actually lower than 1% of the entire sequences is actually mutated as such. And because of the active DNA repair mechanisms, okay, so even though you actually manage to make those mutations, it will be repaired at one point. And if it's too much damage or too much actually mistakes or mutated, then the cell will just decide to commit a suicide. It's just not worth repairing all them and let's die. And so that we don't accumulate any problems. So this is the idea here. Okay, So just kind of good to have this idea that mutation occurs at random. So it can occur at any place. So obviously there's places where you can have, it's more prone to mutation, but they can appear anywhere across the genome. Okay, and the step that actually caused the troublemaker that actually causes those mutations. And I mean, we watch news like even sleep can cause this cancer, like it's just random saying, but like, so the step that actually causes the mutations, usually a chemical, we call them a mutagen. Okay, so obviously they don't have to be a mutagen; they could be simply a UVs and all this stuff. So they are called mutagens. So the question is that only the germline mutations can be carried on to the next generation. So somatic mutations will not be transmitted to the next generation. So this is super important. Okay. And so just kind of let you know that, so you go on the beach and you start like enjoying sunbathing and bathing, and you obviously damage a bunch of your skins and it causes mutations, let's say thymidine dimers after the UV light shining. Okay. So you only kill those, not kill, but like you only causes damage or causes mutation to those kind of surface skin cells, and primarily, and it depends on how deep you're being burned. But like deep in your, let's say, heart or so, don't expect cells in there to be actually mutated. Okay, and what will mutate them though is that if, let's say, like something hurt, I don't know, your sperm or so, and you gave this to your loved one, and then your child, however, will be the one that is being affected. And then all the, let's say the sunbays eventually causes damage to your sperm. And then like this will causes your next generation to be problematic. Cool. So I mentioned this already. So mutation is very rare event, and the mutation even further rarer is that the mutation that actually affect a phenotype. This is even more rare. Okay, so you have to mutate first, and then this mutation has to be consequentially enough to actually causes a drastic change in order to causes a phenotypic change. So those mutations are even rarer. And once you actually have a phenotype change, okay, so then you can use those terms from Naomi's section, say those Miller's morphic definitions, which we will look at momentarily. And this will be the functional phenotypic changes. Okay, so last point I want to mention is that the genes, different genes will mutate at very different rates. Okay, so some are more prone to, some are not. And so those are the concepts. Now let's look at some more detailed terms associated with it. And we're going to go even more details to see which one is which. So the first I want to talk about is the base substitutions. Okay, and in terms of that, this will be the kind of, I would say, the most commonly kind of types in here is that the silent mutations, which we call the synonymous mutations. And this usually refers to that if you actually have this mutation, you only change its base, okay? You don't really result in any amino acid changes. And the second one is called a non-synonymous ones. This is called a missense mutation. So there are 20 essential amino acid, and amongst the 20, okay, and if you originally code for, let's say, one methionine, and then now you suddenly, this base change, you code it for something else, then this is called a point mutation, which results in a missense mutation. You change the amino acid sequence. So within the missense or non-synonymous mutation, you further can be divided into two. So it depends on what kind of amino acid you actually change it to. So if you change the chemical or say biochemical properties of this amino acid, okay, then you are called something called a non-conservative. So I'll just give you an example, say the charges. So for those of you who have taken biochemistry at one point, and you probably would have heard about the two amino acid called arginine and what's the other one that has the very similar charge to arginine? Lysine, awesome. Okay, so we finally have a biochem. I'm a biochem major when I was undergrad. Just anyway, so the lysine and arginine, they are both positively charged. Okay, so if you actually manage to actually substitute your coding changed from a lysine to arginine, okay, so this will actually be considered as a rather conservative in terms of charge wise. So if you happen to be, let's say, in a nuclear localization signal, usually they have a bunch of arginines, and you replace it with a lysine. Okay, and sometimes it will not hurt the property. Okay, so what will change though is that you actually change it to, let's say, a neutral amino acid. Let's say and also like from an arginine. Okay, really like kind of a big in size and positive in charge, and I change it to like a small, like very small, the smallest amino acid, which will be a glycine, and they're kind of relatively neutral as well. And this will be a drastic change in this biochemical property wise, they will be changed. So this is called a non-conservative change. Nonsense. Okay, so just now it's missense, nonsense. Nonsense, literally, it's nonsense. This one stopped to be a protein anymore, stopped to be a full protein anymore. You introduce a premature stop codon to it, okay? And this will most certainly actually causes the shortening of the protein. Okay, so question is, how do we actually detect it? So we'll see biochemical methods, molecular biology methods that can deal with this. And the last one is called a frame shift mutation. Okay, so the nonsense is that I turn a regular coding amino acid, okay, and to a stop codon. So say UAC code for a tyrosine, okay, but UAA, so WA, WAGA, and WGA, those are the stop codon. You turn a UAC tyrosine to a UAA stop codon. So that's called a nonsense mutation. Frame shift. Okay, so frame shift it's also evil. So so you basically alter the entire kind of reading frame. So this will most likely also shorten the reading frame. The coding region because the protein length, because you will very likely through frame shift at one point you will introduce a stop codon. Sounds good, folks? And so again, so I mentioned that you will most likely, but doesn't mean that it absolutely necessarily will do that. So there are really rare occasions. Okay, so you just have this like kind of a perfect kind of a region that you have a frame shift. Instead of actually shortening the sequence, you manage to actually prolong the sequence even longer in terms of reading frame. So like you happen to actually break a stop codon in this case. You like manage to extend the sequence even longer. So this is also possible. In any way, like so frame shift, you change the amino acid sequence for sure. Okay, so the next question is called an indel. Next type of mutation is indel. So this is a larger kind of a scales already compared to base substitution. Just now we talked about point. Now we talk about indels. So it's more like a several bases or a region that is being involved. Okay, so this involves in insertions or deletions. So again, the consequence could be any of the on par with the frame shift wise, but it could also be that you are messing up with a regulatory region. And so by deleting that, you are really messing up the entire expression of this gene. Next one is called a splice site mutations. This is also interesting. So we talked about actually something called intron exons. Okay, so in an ideal situation, the introns will be excised out, and then all the exons retains, and then you express your genes and and get your final protein product. And so there is a regulation of splicing. So it depends on who is being retained, who is not. Okay, so if you manage to mess up the especially the regulatory regions or the mutations that actually causes the splicing regulation to be disarrayed, and as such, you will mess up that's the splicing, and the result could be uncontrolled splicing, and the consequence is severe as well. And in fact, actually, by targeting splicing, Moses for this kind of a work. So they wise in the kind of functional definitions, this will be a loss functions and gain of function Okay, and so I mentioned that if you manage to actually lose your functions, okay, so this will be a loss of functions. So you reduce the function to complete null. So a fancy term to call null mutation will be amorphic. So amorphic, just how functional you are, and amorphic means that you don't have anyone anymore. And so it could also be hypomorph. You just reduce your functions. Okay, so that's hypo means the same thing for hypomorphic, or when you study the osmosis, like the hypotonic. So you're less than the ideal concentration. So gain of function. Okay, so usually you're gaining more function, you get more function than the wild type would have been. Okay, so this could be you have more stronger functionalities, but it could also be you gain some new functions, neomorph and hypermorph. Cool. So the very last point I want to mention, so it's kind of unrelated to this, kind of semi-related, is a lot of the discussions on the discussion board. So SNP versus point mutants, and also whether this is called a new allele or not. So I just want to kind of like introduce my 2 cents, like especially from the first half of the class as well. So as you probably recall, the SNP is when the entire population, okay, so they obviously have a more dominant, shouldn't say dominant, a more prevalent type of a base at a particular, let's say, location, okay, and let's say 90% of the population, 98% of the population has this A in this position, okay, and then if you happen to have actually a population of about 1%, okay, so a little bit more than 1%, that instead of having an A here, you actually have a G here, okay? So because you have actually more than 1%, this is recognized as a SNP, okay, small nucleotide polymorphism, so SNP. So SNP occurs when you have actually more than 1% of the population that has the same nucleotide at this particular spot, and this is this spot, this nucleotide is different from the vast majority of the population. Okay, so the majority of the population has A, whereas your 1%, this Wall Street 1% has T or has G on it. Okay, so this will be a SNP, Wall Street SNP. And so the question is that like, why is it called a point mutation or not? Okay, so the point mutation, as we mentioned already, so their rate may or may not necessarily be more than 1%. Okay, and they're likely less than 1% if you are just a random mutations. And more importantly, the SNP does not necessarily to be caused by a point mutation, but point mutation definitely can result in a SNP. Okay, and so if this SNP happened to be in a gene, on a gene region, okay, so then this will become allele. You can say actually this gene will have at least 2 allele. The first allele is the most prominent one, the vast majority of the population has. The other one is this very SNP at this position. Perfect. Oh, there are questions. So let me read. So if a frame shift result in an earlier start codon, would it also be classified as a nonsense? Okay, so if the frame shift result in an earlier start codon, so you actually had, I would say, have a methionine at a position, okay, and then would that be classified as a nonsense? I wouldn't say this as a nonsense. So there are 2 reasons I want to kind of counteract this one. So the first one is that you not everywhere, not like you can't just by having an ATG immediately you are a start codon. This is wrong. So to qualify as a start codon, you really need to fit in many criteria. So first you have to be a translation start site. Okay, so first to be a transcription start site, you have to fit into certain criteria. So you need to have a promoter and all this stuff. And to be a translational start site, okay, you also need to have for eukaryotes, for example, you have to have many criteria being fit. One of them is called the the Kozak sequence there. So so this really depends on like right before the ATG, do you actually have a particular sequence in front of it. So like if all these are being met, okay, so you probably will have a alternative start site. Okay, so that's the best I can say in terms of the frame shift resulting in an earlier start codon. So alternative start site, this is an annoying situation, but it does definitely occur. So the second question is from Mohammed. So silent, missense, nonsense, and frame shifts are just the effects of the base substitutions. Yes, indeed. That's the consequence of the mutation. Sumia says, for base substitution mutation, is it like one base or multiple bases at a time? Okay, so for that purposes, we for just now, we call it one at a time. Okay. And say like the frame shift, you're just missing one, and nonsense, you're missing one as well. But I mean, like those terms, like again, it's not super strict, so that I don't think exams will be really like kind of a trick you on that. Is it one base or two bases? Like, so you know for sure you can't be more than three because there's only three at a time. But in terms of like for the purpose of this slides, for sure, like and also for your lecture, this is definitely referring to just one. It says, so if it is more than 1%, wouldn't we call the SNP a wild type allele? Okay, so no, so, sorry. So only if you're more than 1% can you be qualified as a SNP, unsNP, okay? And if you are the wild type, then the vast majority of those people, those are called the wild type. So the more majority ones are the wild type ones. Oh, Cherry says that I meant to stop, not start codon. So yes, so in that case, then if a frame shift result in an earlier stop codon, that would definitely I see. So is that a nonsense mutation? Yes, that will be definitely a stop. Sorry, it will be a nonsense mutation. So I just rephrased the question. If a frame shift can result in a earlier stop codon, this will be qualified as a nonsense mutation because it introduced a stop codon. But the question is that so we're talking about two completely different sites. So the site of the frame shift, it's still a frame shift. It's somewhere down the line. At that particular mutation. Okay, so at that stop codon, the newly created stop codon, and which was previously a amino acid, and that location is a nonsense. Does it make sense, Cherry? Awesome. So Mohammed says, is the effect of indel always frame shift? Okay, so this is an interesting thing. Okay, so indel we definitely refer to more than a one or more. Okay, so you have to insert or delete. So, however, the question is, if you actually manage to insert a 3x, or sorry, 3n, or 3 whatever letter you prefer, a multiple of 3 into it, then you do not change the frame. Okay. That doesn't mean that you can keep on adding. I add 300 like bases there, then I can guarantee you, I don't know, you add 300 random, 300 random bases. That's like, provided that none of those 300 divided by 3 is actually a stop codon. Okay, so you just added 100 amino acid to a protein. I can guarantee you the cells will sense what's going on here. Like, this is foreign. They will probably start attacking and realize this is something wrong here. Super. Now let's look at some real examples here. Okay, so genetic codons on the right. So you have to memorize this for exam. Okay. Just kidding. So you don't. Okay, so you're so no, I'm sorry. No. So you will be given this for the exam. Okay. So if it's ever needed. Okay. And so synonymous mutation. So here. So we change from AGA to RGC. Okay, so let's look at the genetic codons. AGA and AGC. Wait, am I right? Just one second. AGA, AGC. Okay, so the mutation shouldn't really be AGC here. It's probably meant to say AGG here. So that will be a synonymous mutation. That being said, if it were a to C mutations, then it would be it would have been a transversion mutations here. Apologize for the typo on the slides. Okay, so again, synonymous mutation means that your amino acid does not change. Okay, so the next one. Okay, so from arginine to a lysine. Okay, so in this case, we knew this is a non-synonymous. Okay, it's a missense mutation. However, because the property of the amino acid is still actually similar, it's all both basic and strongly positively charged. Okay, as such, you can say this is still a relatively conservative mutations. Okay, but it's a missense mutation. So now let's bring some real meat here. So how do you know, like, how do we detect those kind of changes? Okay, so now I want to introduce 3 different assays. So the first one is called Northern blot. Okay, so there's Northern blot, there's Southern blot, there's Western blot. Okay, unfortunately, there is yet to have a Eastern like blot that is being popularly used. There is something called it, but we don't really use it often. Okay, so we're just literally just playing those names. Southern is just the guy's name. Okay, so Northern blot is for RNAs. Okay, and when you carry those mutations, okay, and you want to detect it, so RNA, you run a Northern blot, and then you use some, let's say, fluorescence probe, or use radioactive probe to probe it, and then this will be a method to detect it. So in terms of the length of the mutations, okay, so the mutation will still actually retain. So it doesn't matter synonymous or non-synonymous missense ones, you will retain the same length of the sequence. Okay, in terms of the RNA sequence, it will be retained to the same. That's to say, if I run Southern blot, which is dealing with the DNA, their sequence should still be the same as well. Okay, and it shouldn't really change the length of the product. The next thing is called a Western blot. Okay, Western blot is to run a protein, your final kind of a product of the protein. So all these gels are actually denatured. So for the protein gels, and ideally missense mutation, just this one base. Okay, so it will just change one amino acid at a time. So likely it will not change the size of the protein product. Okay, that being said, I would so it's to the level that it's not easy to be detected. The change in size will not easily be detected by the gel itself. Question being though, like, so what's really being changed here by missense mutation, especially not a conservative ones, you would definitely change the properties of the proteins. Okay, so the actual consequence to it is definitely more than just to change the size itself. So the size does not necessarily change, and it will probably not change that much, but the biochemical properties will be changed. But how does it actually reflect on the Western blot then? So remember we said that because this is more so a size-based, because it's it's denatured gel, so very likely the Western blot won't change in size either. Okay, I do want to kind of warn you a little bit for those of you, especially you have lab experience. So things like phosphorylations, and for example, they will definitely change the pattern of the gel migrate. But this is again outside the scope of this class. Okay, so let's go back to the question board. So Mohammed says we're not required to know if the missense mutation is conservative or not, right? So the definition itself should be sufficient. Okay, so you will not be tested. It is not a biochemistry class, so it won't be tested on the amino acid properties. That being said, it's a fair game though if the question stem actually specifies so which will causes a change to its biochemical property and of this amino acid, then you should know this is not a conservative one. So Maya says, what's the differences between northern and western blot? Yeah, so I mean, this is annoying. People are, biologists are funny. They're playing with those names. So northern is for RNAs, and southern is for DNAs, and western is for proteins. Okay, they all separate by charges. Sorry, they all separate by size because the gels are denatured. I'm talking about the proteins, westerns. So are mostly separated by size only. Incidentally, the faster you migrate, okay, so usually you put a charge, okay, so usually your nucleic acid and your proteins are, in this case, renning are both negative charge. So you will put a positive pole in the bottom and negative pole on the top, and they will start migrating based on this pattern. So obviously, the bigger you are, so size-wise, the bigger you are, the slower you will migrate. Okay, so the third one, nonsense. Okay, so nonsense, it will introduce a stop codon. So again, so let's go back to the Cherry's question just now. Like, so if this at this very site where a amino acid is being changed to a stop codon, then this will be a nonsense mutation. Okay, so nonsense mutation, you can pretty much be guaranteed that the proteins will be shorter, but the RNA itself or the DNA, the nucleic acid itself, will not. Okay, so the mutation occurs on the DNA, but the RNA, the transcribed things, you transcribe it as the same length as yes. Okay, so all of those products, by the way, they will be all the first three. Okay, so they will be transcribed as yes in lengthwise. But protein product-wise, there will be a difference, significant differences. For the nonsense mutations, you definitely will change the migration pattern because you shorten the proteins already. You introduce a premature stop codon. And remember, I said that the faster you migrate, the smaller the size you are. So as such, you would expect the gel to be running faster. Your product to be running a bit faster than the wild type version. Yeah, anyway, so I mean, I should go forward now. My computer froze. So in terms of missense mutation shouldn't change either way. So they shouldn't change the transcription product. They shouldn't also change the protein product either. Oh yeah, that's working. So frame shift. Okay, so frame shift is kind of interesting. So frame shift, theoretically, it would say that I add one base, okay, or two into it. And or like, I don't know, for just like for the purpose of this slides, you should add one base, okay? And so that also goes to somebody asked question just now. So do you refer to one or two? So yeah, we're multiple, just one base. So realistically, theoretically, there is a change in length in transcribed product because you're shortened by one, okay? The question is that can you actually resolve that one base pair change on a 3,000 base pair long gene? You can't. Okay, so so relatively on the northern blot on the RNA transcription mRNA gels, you won't be able to see the transcript size drastically changing. Okay, there is a 1 + 1 or -1 change, but not that drastically changed. What differs though is the Western product. Okay, so theoretically, you can go both ways. You can go bigger and smaller, okay? But definitely you will change because of the frame shift. Sounds good. So that's it. So that's all the first 4 types of single kind of a base substitutions ones. Now let's look at mutations in the regulatory regions, okay? And say, like say, if you are mutating in the promoter region or in the terminal sequence, termination signal size, okay? This will actually mess up the transcriptions, the expressions of these genes, okay? So, and even more, you can actually be this splice donor acceptor kind of site mutations, you will write or mess up the splicings. So both will actually mess up the expression of these genes. So we already mentioned this, and this is the second time. If we actually base that on the thing called a Miller's morphic definitions, then if you have a loss of function, okay, this will be the final protein product will actually lose a certain percentage of the function. This will be called a hypo, so you're less morphic mutation. But to the very extreme, you lose completely everything. Okay, this will be a null mutation, amorphic mutations. I just want to quickly say that later on you'll probably heard this term called CRISPR, okay, in this class or everywhere now in the news. So CRISPR, one of the things Toronto really trumps at is something called a CRISPR-based loss of function screens. They use a CRISPR library. Basically, they disrupt the functions of a gene, okay, and so causes a loss of function of a particular gene. So, and then if you manage to actually lose the function of one gene at a time, okay, and then you try to associate that with a particular phenotype you want to observe, then you manage to actually figure out, oh, this gene is actually causing this phenotype. Kind of a classical reverse genetics approach. The very last one we want to talk about is called a gain of function mutations. So this happened to be also my PhD thesis before, and which basically a gain function is a mutation in the gene. Okay, it will causes a gain of the function that you kind of originally have, and so it will have more function of it. So it will be like hypomorph function. But what's really interesting is that you can actually gain something that is non-related to it as well. So it's a tumor suppressor, you manage to actually gain something like a gain oncogenic function. So this will be a neomorph function to it. This will all be gain of function. You gain the function compared to the wild type. So go home, and when you go home, and I want you to actually write out this kind of yourself and think about, like, compare the null mutation, okay, and the gain of function mutation and the loss of function mutations. See that what are the differences and what are the similarities amongst them. All right, now let's look at a question. Just give you 1 minute to work on this, and then we try. Awesome. What's the verdict? Perfect. Okay, so that's perfect. So on the exam, okay, so the fastest way is to get what you want and then move on, okay? And if you have time, if you have the leisure of time, okay, then you should really go into them and see why the other ones is wrong. But when you practice, definitely know why the other ones are wrong, okay? So correct answer is definitely B. This is basically just know this statement is right. And neomorph sorry, just one second. Neomorph will result a similar product, a similar level of product being expressed as wild type, but their functions are very different. Okay, so they are more evolved. And so hypomorph is the exact opposite. So that's choice A. And C, amorphic, okay, this will be the exact definition of null mutation, okay? And so they're on par with each other. And the gene deletions will be the same as null mutation function-wise. Question number 2. Whoops. So this kind of question, there is really no tricks, but just go to the table, which will be given to you. Okay, so I think but I want you to think about it. Is everyone okay with choice B? CAC to CAA. Perfect, perfect. So CAC is a histidine and CAA is what is that? It's a glutamine. Okay, so it's a different amino acids. Okay, so just a few things I want you to kind of know that. So what is that genetic code? I know that's kind of a high school stuff, but it's annoying when it comes to your genetics class and you realize, oh, hey, what is that? So this codon, okay, we're referring to the mRNA sequence or the tRNA sequence. This is the thing I want you to think about. And is the sequence the sense strand of the DNA, the mRNA sequence, and the tRNA sequence? Which one is which? I want you to think about it and just pay attention to it. So on the exam, probably we won't trick you like very hard on that, but there's no guarantee. If we do, like, I guess it's a fair game as well. So I'm just saying that so there are three things you probably, three sequences that are involved. The first one is the DNA on the DNA coding strand. Okay, so what is the sense strand sequence? And the second sequence you can think about is the mRNA sequence. And the third one is the tRNA sequence. So which one is which? What is the one that of the genetic codon? Anyway, we'll talk about this after we come back. Okay. It's the RNA. So yes, so this question is exactly as is. Okay, so this is the RNA. How do we know that? This is the U here on the choice. This is the RNA sequence that we're dealing with. This is exactly the genetic codon we're dealing with. Cool. Next one. A researcher discovered a mouse with nonsense mutation. Again, I'll just ask you to read it. What's the verdict right now? Awesome. Okay, so it's a nonsense. Okay, if it's a nonsense mutation, you prematurely introduce a stop codon. Okay, and the final wild type, final protein product will be less than in size compared to the protein. So what would happen to the Northern blot if I were to run the RNA sequence? The mRNA. Same. Perfect. Okay, so that's it. That's good. That's good. Perfect. Okay, so the final item of today is transposable elements. Okay, so let's talk about this. So again, so we're going back a little bit again into the classical genetics about the discoveries. And so can't remember this lady's name, but I like this Mike, I think it's Mike Clint, Clintable. And so this is a very, very classical experiment that's done for the discovery of transposable elements. And this is done in the corn, in the kernels. And so not going to repeat what the class is talking about, but just know that there are two big classes of transposable element. One is called the retrotransposons. Okay, this is commonly known, and this is basically it uses RNA precursors, and they integrate it into the genome. Okay, and there's also long-terminal repeat types. There's also non-long terminal, non-LTR kind of a types transposon, retrotransposons as well. Okay, and they can make many copies, and they are very replicated. Okay, so they can make many copies in the genome. And so the examples in here would include line signs, and which we talked about today, and those are non-LTR type of retrotransposons. The other one is called a DNA transposon. Okay, so transposase enzyme removes elements, and they basically place it into another region of the genomes, which we'll talk about the maze momentarily. And so there is only one copy that it kind of moves. So they are not replicated. So this is the differences between the two. So on the left-hand side, the retro ones, they will use RNA precursor, and they are replicated. And so they will have multiple copies. The other ones, the DNA ones, they will only have one copies. Yeah. So historically, transposable elements is thought about like junk. Okay, so they're just like randomly jumping, and they're not. So they are very important for the regulations, the silencing of a particular genes. And in fact, before CRISPR technology is actually out, and many people actually uses a technology called a sleeping beauty. So they use transposon systems to actually screen for the functions of the genes. Okay, so they basically use the transposable elements ability to randomly insert and thereby disrupt the functions of a gene region, and as such to figure out to perform screenings, reverse genetics screenings. That you have DS and AC presence in the middle of so there are two versions of mutant alleles of this state. Okay? So if you have ACs or DS in the middle of the C genes, the result is a colorless version. The fact that AC itself can jump in and out, okay, autonomously, and as such, this AC presence in the middle of the gene will be spotted, okay, because it can jump in. Once it's in, you disrupt the color, it's colorless. Once it's out, it's purple, so it's in and out, okay, it's spotted. And for the DS, okay, so when it's in, you are disrupting the colors, it's colorless. When the AC is nearby, this is the only scenario where it can actually, DS can actually jump out. So when AC is in the nearby scenario, and this will causes the change. So I did a bad job just now explaining this entire chart. So let me just repeat it again. So I have a wild type, okay, so that coded for this purple color. I'm sorry, just now it's coded for the pigment color. And when you have something inserted into it, this causes a mutant allele. Okay, so the mutant allele can either insert DS or insert ACs into it. So inserting any of the two, either of the two, will disrupt the color into colorless. Okay, the question is that can you remove that element out or not? If you can remove that element out, okay, the DS or the ACs out, you become a spotted ones. So the question is, can you jump out or not? So AC is autonomous, so it can jump in and out on its own. So the result is that putting an AC here, you will always have a spotted kernel. And the DS, okay, only can jump out when AC is present. So as such, only when the AC is present can the DS be resulting in a inserted allele causing a spotted kernels. And AC is not there, then it's stuck there for good. It's colorless. So Mia says, when AC is nearby, DS can jump out. But since AC is actually inserted, it forms spots. Is not. Boo, I'm not sure I understand the question. So when AC is actually inserted into the C genes, it's not actually inserted. So basically, when AC is inserted in the middle of the C genes, okay, it will disrupt the C gene expression, and it causes colorless pattern. But because AC is autonomous, it can jump out as well. As such, it's spotted. So when DS is inserted, it causes colorless, but it'll only become spotted if AC is nearby. Does it make sense, Sumia? In other words, if you insert a DS, it will get the second scenario colorless, okay? But once you have AC nearby, it will be a spotted version. Oh, I see. So is the AC like inserted? Okay, I got it. Yes, no, AC is not inserted. Okay, so AC is present in the genome. Yes, so here when I say inserted, I meant to say it's inserted into the middle of the C genes, which is the last scenario. Yes. So Jeff says, so it will be spotted if AC is nearby or inserted. Correct. Yeah, sorry about the confusion. Yeah. How's that one? Cool, let's do it. So how many people think A is correct? If you think A is correct, just type A. So forget about the question stem. So don't worry about it or not. I'm just individual items. If you think A statement is true, then type A. Awesome. Okay, that's indeed the definition of transposase. Okay, transposase, not definition, but a function of the transposase. It will cut target DNAs. Okay, so it will be used for this jumping as well. And so B, we think it's right. Perfect. So B is correct as well. Question lies in C. So what do you think C is right or wrong? If this is right, then it should be a none of the above. Wrong. Okay, so we have one person saying wrong. What about the other people think? So the answer is wrong. Okay, so transposition does not necessarily result in the deletion of the mobile elements. You can retain it there. Okay. And in fact, like, so you can actually have in a particular region, you can actually have a tandem repeat of the same thing over again, and multiple times in the same region. Cool. So the correct answer is C. So C is not necessarily true. Cool. Perfect. So I think I got one more questions. And so is everyone okay with the last question? This one right here? Perfect. Thanks. Cool. Final question. So everybody obviously have linked this to the to the corn example just now in the maize corn example. So the question is, what's going on here? Anyone want to volunteer for the choice? A. So Cherry says A. So I guess you can either choose A or C. But which one is which? I'll tell you the answer is C. And let's think about it. Why? I'll let you to think about 1 minute, and then we'll talk about it together. Perfect. Okay, so let's talk about it. So there are two things we need to talk about. So first one, who is the background? Okay, so for this very particular crop in question, okay, it's a yellow background with occasional red spots on it. Okay, so it means that the original, let's say the ones that actually has the transposable elements, okay, and what would be the color for it? It's a red background. Sorry, it's yellow background. And then by removing or inserting a particular thing, okay, and revert it back to the original color, you revert it back to the other color. Let's go back to this example here. So basically, let's go back. Okay, here. So the wild type is the red one. Okay, so if you don't have a transposable elements in it, okay, it'll be red color. And once you, sorry, it will be a yellow color and the background. And once you actually excise it, it will be resulting in the spotted genes, spotted phenotype. So this will be the red. I'm not sure that makes sense. Hence the C answers. The wild type will be the red. It happened to be this gene, so the original gene is actually the yellow form. Correct. Yes. Right. So there is a question here. So Mohammed, so just a general question. Some cells have AC in their genome and others don't in the maze examples. And is that correct? So yes. So you have ACs. Okay. So in the ACs, it has to be in the vicinity of the DS. And because it's a DNA version, so you only have it once. You don't really have it everywhere in the genome. So you only have in one location. And if that location is happen to be next to an AC, then you have the effect. And Sumia, I was referring to this, yes, the excision actually causes the spots. And Jeff says, in the corn example, wasn't it insertion? So are we talking about the question I'm showing right now, or the slides? Can you mind just elaborate the question a little bit? Do you mind just turn on the mic and elaborate a little bit more? So in the corn example, wasn't it insertion? Like AC was inserted in the wild type. Yes, exactly. So that's what Sumia was referring to. Yes. So basically, she says that there is spots in the example since AC stays. This is when AC stays in, and then when they jump out or excised out, this is where the spot actually occurs. Wasn't sure that I answered the question. Just go back to here. So does that answer the question, Jeff? No. Okay, cool. So let's go back. Yeah. So I was just wondering, I'm just going to stop recording. So I'm just wondering if you can kind of elaborate, like which part is confusing.
Oh, I'm not getting this properly. So if red is the wild type, a mutation causes it to be yellow, right?
Yeah, sure. Let's go through this question again. Okay. Yeah. So, so you start with, let's see, here. Oh, yeah, it's right here. So it says this one has red kernels. Okay, a variety of corn with red kernels. Okay, so the gene itself then is red. And so red pigment genes. Okay, so your wild type is the red color to begin with. And so now this person actually identified a corn kernel which was yellow. Okay, and this is already a telltale sign that something got inserted, messed around this red pigment genes. So it got inserted. So the formation of the yellow is caused by the insertion of the transposable element, which I call it T here. So when the T is present, yes, the plant is yellow. And now the question is, what if actually the T jumped out? What's going to happen? You resume the red color. And that's where the red spots comes in.
But if the T jumps out, doesn't it go back to being red again?
Yes, it could very well be if it were a autonomous ones. If it were a AC,
okay. But this one is a non-autonomous, right?
Well, no, it doesn't have to be non-autonomous. Like, so it can jump in and out. You feel free to do that, but it's still it would still not change the pattern of being spotted, because you can't synchronize everybody like in and out, right? Like so the ones the regions where it jumped out, it will still be red. And that's where the mosaic actually comes in. It's because only certain regions the AC jumped out, or where the transposable element jumped out, and certain regions that didn't, and that's where the yellow retains.
Okay. Yeah. Thank you.
Let's turn this question around a little bit, and let me think about it. Yeah, so why don't we say that some other dude like find a red kernel with yellow spots. What's going to happen?
Wait, so is the wild?
I'll tell you that this is the I mean let's be rigorous and specify this is actually
Wait, so
it's called red pigment gene. It's a red. Wild type is red.
Okay, wild type is still red.
And the dominant is red as well.
Okay.
But yet this person has a red background with yellow spots. Then your insertion theory might work. Those spots that actually has yellow spots on it, this could be the result of the spots where the elements actually comes in.
So it's the insertion of transposable element into the into the red pigment gene, right?
Correct. Yes. If it's yellow spots.
Yeah. Okay.
Yes. So the bottom line is that its spotted pattern comes as a result of the gene jumped out. So you returned to the dominant version.
Okay.
Cool.
All right. Thank you.
No worry. Yeah. Does it make sense?
Yeah, makes sense.
Cool. Thank you.
Bye.
All right. Have a good weekend. Yeah.
You too.